Hello, and welcome to this webinar on opportunities to transform medical research so that it benefits and protects both people and animals. I'm Dr. Hope Ferdowsian, co-founder, president, and CEO of Phoenix Zones Initiative, and a professor in the Department of Internal Medicine at the University of New Mexico School of Medicine. PZI is a global nonprofit that uses medical and public health expertise to solve problems for people, animals, and the planet. PZI's programs focus on education and outreach, research and analysis, and advocacy for changes in policy and practice. We advocate for international, national, and local policy changes that require international and government agencies to abide by principles that safeguard people, animals, and the planet against exploitation. We're especially focused on addressing publicly funded activities such as food policy, medical research, public health priorities, and international development. In this webinar, we'll focus on opportunities to transform medical research, particularly in the U.S. We'll start with some big questions about the health of the nation and then move on to how scientific and ethical innovation can benefit both people and animals. To talk seriously about transforming medical research, we need to start with a key question. Why aren't Americans getting healthier? That's a question asked of the former NIH director, Dr. Francis Collins, in an NPR interview. And it's an important question since taxpayers task the NIH, FDA, CDC, and other agencies with improving the health of the nation. With respect to the question of why aren't Americans getting healthier, it's a complex question with a complex answer. But a big part of the answer is that there's a lack of focus on prevention. The two biggest killers of Americans are now heart disease and cancer, largely preventable diseases. The top killers of children in the U.S. are firearms and motor vehicle accidents, also largely preventable. And when researchers have examined actual causes of death, sometimes referred to as behavioral causes of death, poor diet has now surpassed tobacco as the largest preventable cause of death, including from heart disease and cancer. Actual causes of death from diet, activity, tobacco, and other behaviors are influenced by preventable social, economic, and environmental determinants of health, which are determined in part by legal and political priorities. Determinants of health can include access to safe homes, workplaces, and communities, access to education and opportunity, access to clean air and water and nutritious food, healthy social interactions and relationships, and the legal policies and political priorities that shape these social, economic, and environmental conditions. Social, economic, and environmental determinants of health are now so important to medicine that we have medical billing codes for them. Unfortunately, national spending does not reflect research needs. When NIH researchers have compared risk factors for death with NIH spending, they found that although 57% of deaths are attributable to any top 10 risk factor for death, only 34% of NIH projects address these issues or areas. These disparities are especially notable for dietary risk and tobacco use. In addition to there being a lack of spending on prevention and prevention research, there's also a problematic reliance on animal experimentation. Before medical interventions ever reach clinical trials, there are huge investments in discovery and development and preclinical research, now also called non-clinical research. Let's look a little deeper at the drug discovery and development process. It starts with discovery and development, where animals are used in countless numbers. Once researchers identify a promising compound, they conduct further experiments to gather information on how it's absorbed, distributed, metabolized, and excreted, and any information they can gather on potential toxicity. Researchers also look at a compound's potential benefits and mechanisms of action, and the best dosage and the best way to deliver the drug, such as by mouth or by injection. Once a drug has passed through discovery and development, it moves to a preclinical phase. Preclinical research typically has historically required tests on two non-human species, typically rodent and non-rodent tests, such as dogs or non-human primate species. That changed with the passage of the FDA Modernization Act, which passed in late 2022. And that's good news, and I'll tell you why in a moment. Once companies get past the non-clinical or preclinical research phase, after they've spent a lot on discovery and development, they must submit an IND, an investigational new drug application, so the FDA can review their preclinical data. Sometimes phase 0 studies may be done. Phase 0 studies use only a few small doses of a new drug in a few people, but usually clinical research starts with phase 1 studies, small studies of 20 to 100 healthy volunteers or people with the condition to test safety and dosage. About 70% of these studies move forward to phase 2 studies. Phase 2 studies include hundreds of people with the disease, and they may take months to years to test efficacy and side effects. About a third of these studies move forward to phase 3 studies. Phase 3 studies include hundreds to thousands of people with the disease over about 1 to 4 years to test efficacy and adverse reactions. About a quarter of these studies move forward. Typically, after a phase 3 study, FDA reviews company data for a new drug application, or sometimes referred to as an NDA. If the FDA moves forward with the new drug application and it's approved, that is followed by release of the drug and post-market surveillance. Phase 4 studies may be conducted, and they include thousands of people with the disease to further test safety and efficacy. As you can imagine, relatively few drug candidates make it to market, and the system is intentionally set up this way. So I mentioned that preclinical or non-clinical research, before it gets to any human clinical research, may change with passage of the FDA Modernization Act. And that is really good news for people and for animals. Animal bioavailability data does not correlate well with human bioavailability data. When I mention bioavailability, I'm just talking about the amount of a substance, such as a drug, in the circulation, in the bloodstream, that is active and able to achieve its intended effect. For example, if you look at the bioavailability of a range of candidate compounds in dogs, they do not correlate with human bioavailability. The same is true for non-human primates and for rodents. Dogs, non-human primates, mice, and rats all belong to species commonly used in medical research. You should be able to draw a straight line through this graph to see a meaningful correlation between animal and human bioavailability, but that isn't possible, as you can see here. And as a result of the dramatic disparities between animal and human data, we see high rates of false negatives when a test is negative and wrong, and high rates of false positives when a test is positive and wrong. And that means increases in the risk for adverse events in humans, whether they're in clinical trials or in the larger patient population, and delays in drug discovery. It's important to note, though you probably already know this, that when we're making daily clinical decisions in medicine, we don't rely on animal experiments. Physicians follow a hierarchy of medical evidence in recommending lifestyle modifications, medicines, vaccines, surgical procedures, and other interventions. The top level of evidence includes systematic reviews and meta-analyses, filtered information that summarizes unfiltered information from randomized control trials where participants are randomly assigned to one arm of a study or another, cohort studies where we observe populations over time looking forward or backwards, and then we may consider case-controlled studies where we look at cases and common exposures. That's how we know, for example, HPV causes cervical cancer, by looking back at cases for common exposures. And if little evidence is available, we might consider more anecdotal case series and case reports. We also consider generalizability. Is the research population in the study or studies similar enough to the patient or patient population I'm treating? That's why it's best to have studies, human clinical studies, or other types of studies involving people that are diverse and representative of the patient population. For example, if I have a patient who's a 65-year-old woman with heart disease, I'm not going to reach for a study of only men with heart disease. Similarly, if I have a patient who's a 75-kilogram man with heart disease, I'm not going to reach for a study of mice who weigh 30 to 40 grams and have a range of anatomical and physiological differences from the person in front of me. This hierarchy of evidence I've shared is also important to how we evaluate animal research more broadly. When physicians and scientists have analyzed the sum of published animal studies through systematic reviews and meta-analyses, they've shown that the predictive value of animal studies in determining clear evidence of human benefit is very poor. I'll tell you about a few examples. Daniel Hackman searched the 7 leading scientific journals that regularly publish animal research: Science, Nature, Cell, Nature Medicine, Nature Genetics, Nature Immunology, and Nature Biotechnology. He selected the most cited animal research to see if it translated in human studies. He observed that no animal study was reported as negative, pointing to the problem of publication bias, where positive but not negative results are more likely to be published. Dr. Hackman poured over 2,000 studies, and 76 met his selection criteria for his review. Of the 76 animal studies, 37% were replicated in human randomized clinical trials, 18% were contradicted by human randomized clinical trials, and 45% were untested. 11% were subsequently approved for human use. Does that mean that animal research was necessary for the development of even that 11% of human-centered treatments? No, it just reflects the pipeline of development that I shared with you earlier, from discovery and development to market. This study by Dr. Hackman didn't take into account post-market surveillance, where some drugs can be relabeled with warnings or pulled from the market. Elsewhere, Pablo Perel and his colleagues have used systematic reviews and meta-analyses to compare treatment effects between animal experiments and human clinical trials. For example, they examined 6 areas where clear evidence of human clinical effect was available, and half of the human and animal studies were concordant, so they matched, and half were discordant, so it's almost like the flip of a coin. A more recent 2019 systemic scoping review in the Journal of Translational Medicine showed that translation success rates were also unpredictable. So why does animal research fail to predict human outcomes so frequently? Well, one of the problems is that animal experimentation presents many threats to external validity, that generalizability question I posed to you. Humans and other animals develop different diseases, and disease progression differs across species. For example, other animals don't experience heart disease as humans do. Certain cancers aren't found in other animals in the same way that they're found in humans. Humans are the only animals to have permanent breast tissue, for example. An individual's health status, whether we're talking about a human or an animal, also matters to how they develop disease. More on that shortly. Animal experiments don't represent the diversity or heterogeneity we see in the human population. There are also differences in outcome measures. For example, how would we adequately assess for side effects such as headaches and other subjective symptoms in dogs in laboratories? Although dogs likely experience headaches, they have no clear way of telling us. Headaches are a common side effect of medicines in clinical research and in patient care. Lab experiments involving animals also don't account for human epigenetic effects, the huge effects of behaviors and the environment from birth to death on genetic expression and therefore disease risk. And lab experiments don't account for human genetic variability as more modern technologies can. Animal experimentation also fails because of problems inherent to animal research. The rigor of animal research is nowhere near that of human research. More deeply, the acute and cumulative trauma of laboratory life also has a profound effect and interferes with the interpretation of experimental results. Animals face a range of unnatural stressors, from forced breeding and family separation to prolonged captivity and isolation in laboratories. Like us, they feel fear, anxiety, and pain, and they fear being hurt and killed. In humans and other animals, these unnatural stressors lead to changes in the brain. For example, post-traumatic stress, depression, anxiety. These unnatural stressors also lead to changes in the heart, the immune system, and virtually every other organ system. And these confounders can't be reliably teased out from the experimental outcomes of interest. In the laboratory, there's no way to sufficiently control for these confounders, not even with welfare advancements or standardization procedures, because we're talking about individuals who are sensitive and capable of complex joy and complex suffering. They aren't simply tools. Because of these and other concerns about animal experimentation, the field of toxicology that involves safety testing has moved toward a more evidence-based approach in toxicology, reflected in something called Tox21, Toxicology for the 21st Century. Toxicology is essentially moving from a linear testing strategy to an integrated testing strategy. At each step, population-based data and human exposure information are considered, as is the question of what data are really needed for decision-making. As a result of the availability of superior methods, experts in toxicology expect a fairly complete shift away from animal methods. And although Tox21 has primarily focused on chemical testing, there's also a move in this direction in the field of pharmacology. The need for more human-relevant research is driving a move to non-animal methods, also called new approach methods. Here are just some examples. Human organs on chips are microfluidic devices lined with living human cells that can be used for drug development, disease modeling, and personalized medicine. They were originally developed at the Harvard Beas Institute. The company Emulate has leveraged organ chip technology to mimic human organs in vitro, enabling faster, better, and cheaper drug development and insights into human health. Systems like Epiderm by MatTech Life Sciences are three-dimensional cell models, bioengineered tissues for chemical, pharmaceutical, and skincare product testing. And Mimic Modular Immune in Vitro Construct is a surrogate human immune system that enables researchers to test the immune response to a vaccine, a biologic, another drug, an environmental irritant, cosmetic, or another product directly in a human-based system. This system accounts for genetic variability and differences within human populations. An in silico design involves virtual investigation with computer models and virtual reality. In silico methods have been used to predict therapeutic responses to COVID treatment strategies, HIV treatment strategies, various surgical approaches, and more. The list of possibilities here is really endless. The global market for new approach methods because of advancements in these and other areas is growing, and it's expected to reach $1.65 billion at a rate of 11.6% through 2023. But that's still just a fraction of the research and development market. That's why we must also push values and ethics forward. In human research, we've seen how higher ethical standards have made human research more ethically and scientifically rigorous. Egregious, unjustifiable human research practices led to the congressionally mandated Belmont Report, which established ethically guided protections for humans in research. And these protections stem from the principle of justice and a central tenet in medicine: do no harm. My colleagues and I have written about how the time has now come for a Belmont Report for animals using similar principles, both because that's what's right and because better ethics leads to better science. In human medicine, we saw how improvements in ethics led to more scientifically rigorous research, and the same could be true for other areas of research. But in closing, I'd like to leave you with one thought. It wasn't necessarily scientists or even doctors who pushed for the Belmont Report and improved protections for people. It was the public, and Congress responded. If you'd like to learn more about PZI's efforts to produce a Belmont Report for animals toward better ethics and better science, check out our website at phoenixzonesinitiative.org or transformmedicalresearch.org. Thank you so much for listening.